All right, welcome back. This is the Chapter 13 discussion on host defenses. This will be our last presentation that we'll be doing. We'll be moving into review sessions and all sorts of things that try to prepare for exam 3 and the final and the lab practical and all that good stuff. We're getting down to the end there, which is good, but I don't want to get too close to the end that we scramble. So it's good timing. I'll try to get all of Chapter 13 this time, and hopefully you've had a chance to review the videos. That's helpful in terms of getting through the slide presentation that I have for you. It took a little bit. I've had to do this a couple of times. I've had some technical issues. I try to embed some of the videos in the presentation, and that was a disaster, given that's McGraw-Hill's proprietary video, and they had put a protection scheme on it or something. Anyhow, we learned it doesn't matter. I was able to prepare it. I put it on YouTube as a private so that those with a link can see it. That way I go along with all the laws, and the materials that I'm presenting here, hopefully it'll make this section really clear. This is an important chapter. This kind of ties together the concepts of immunology. So let's get into it. We're going to go after the very third line of defense, comparing terms like antigen, immunogen, and epitope. We're going to be going over those again, the vocabulary, but this is a conceptual part. It's abstract. Immunology is abstract. I took immunology at Duke University. What I did, we're allowed up to 30 hours, I believe, with the universities outside NC State, where I was going at the time. And so I went to Duke for my immunology and UNC for my statistics. And I just suggest that always look at see what kind of, the way the game's played. And I could pay state tuition, go to Duke. You're gonna, you can bet you I'm going to do it. But what I did is I audited a class and then took a class. So I would come in for a medical immunology for the audit, and for the 4-credit class was a regular immunology, a full immunology type of class. And it really worked out well. So I mention it just as a suggestion for you to think about whenever you get into your programs. Look at what sort of flexibility you might have in getting your degrees. 4 stages of specific immune response. We're going to talk about that, and cell markers, or the license or paperwork that one has to have in order to have the proper responses. And the key player in that is the major histocompatibility complex, and we're going to talk about that. Probably not so much in 2 sentences, but we'll try to do the best we can. The type of immunity we're going to be talking about, it's adaptive immunity. It's acquired only after an immunizing event. Now, I know that can sound kind of strange, but anytime you get exposed, that's an infection, but that is an immunizing event. And so after that, hopefully you'll have long-term immunity, and you'll be able to deal with it the next time, of course, if you survive the first one. So we're going to talk about B and T lymphocytes, talking about selective processes that prepares them for reacting. And immunocompetence, the ability of the body to react with countless foreign substances. If you're immunoincompetent, then you're not able to do that. And so again, immunocompetence is an important term. The consequences of antigen-antibody binding. I just want to kind of state it out there. I like to tell you who done it, and so there's no mystery. The butler did it right up front. So agglutination, antibodies cause antigens, microbes to clump, and it's a very important test. We use that to determine blood types by clumping the agglutination types of tests. Other words that we're going to talk about are opsonization and phagocytosis. Opsonizing is covering bacteria, whatever the foreign substance with antibody, and that's the process of opsonization. And that enhances the phagocytosis because the FC portion, or the very bottom of the antibodies, are conducive for these types of things to stick to, something to bind to get. So the antibody is sort of like a steak fork, and the end of the steak fork is what makes it easier for the phagocytic cells to grab hold of, and what it's binding to is whatever you stuck the fork into, so that kind of goes along with it. Neutralization causes things to stop from binding. It blocks them from mucosal surfaces. That's why if we had these immunization type of things they're talking about for the virus, we wouldn't worry so much. Activation of complement, that's a cascade event. It's sort of like starting a cascade of events with dominoes. And once it started, it ends up lysing cells. Inflammation, it's important to get the immune cells in there, but it also is painful and it does host damage. It's a kind of a give and take. And then antibody-dependent cell-mediated cytotoxicity, where antibodies attach to the target cells, and then that causes a destruction by non-specific immune cells. So that's in a nutshell sort of the consequences and why we're studying these sorts of things. And I hope you've had a chance to review the immune response. I think I set that up twice. It's, again, I just can't go over enough. It does a really good job of getting you sort of focused on the important things. And that's half of it in immunology, or any subject matter for that point, is noticing what's important to retain and what's not important. And I try to point those things out. Antigen, that's important. Molecules that can be seen and identified by the immune system. So antigens have certain characteristics we're going to talk about. They provide or invoke or provoke a response that's called immunogen. So not all antigens are immunogenic, or some could be poorly immunogenic or poor immunogens. And that could be part of the characteristics. They could be covered with a glucose or a mucus or something, and that just doesn't do very well for immune response. But if there are protein polysaccharide molecules on or inside, those are antigens that usually provoke a pretty good response. Any exposed or released protein or polysaccharide potentially is an antigen, even those in our own cells. But our own antigens don't usually provoke immune response, because at some point of our development we kind of push a button and say, okay, tear everything, or what we're going to do is make everything from now on non-self. In other words, everything that has sort of been tagged with our MHC and our barcode, don't touch it. That is ours, and no need to create an immune response. After that, then anything that we see gets the immune response. Characteristics of specific immunity: antibodies are produced. It's like the chickenpox virus or measles virus, and we get that specificity, and it'll go after those. It can be totally different things, and we make a set of immune responses to each, and it just adds up. Memory is a very important characteristic. Higher mammals have it. We're programmed to recall, pull out of storage, out of the closet, responses that we've already had, and the ability to get to it and implement that is much, much faster, and I'll show you later on what I mean by that. Stages of immunological development and interaction: lymphocytes development and clonal deletion. And I'll just go ahead and tell you, clonal deletion sounds really wow. It's not. All it's meaning is that any part of the immune response that could potentially damage our own host or whatever, we get those deleted. And so it's a population of troublemakers potentially on the antibody or T cell side. We just get rid of it. We don't need it. presentation of the antigen and clonal selection. In other words, of all the things that we make, which ones actually push the buttons that we need, and those are the ones we want. The rest of them, we'll get those through clonal deletion. Get rid of them. We don't need them. The challenge of B and T lymphocytes by antigens, which that's the challenge. It's always a battle. T lymphocytes response, cell-mediated immunity, and B lymphocyte response. So just know that T lymphocytes are typically the cell-mediated part of the immunity, and the B lymphocyte response is the activity of antibodies and the production of same. And so the T lymphocytes also help regulate and provoke the B cell or the B lymphocyte response. And so I guess that's it. No, I'm just joking. We got a lot more to go on. So the immune response, all lymphocytes arise from the same basic stem cells. So stem cells are types of cells that have the keys to all the aspects of the genome, so they can turn on whatever gene processes that they want, and they can be any cell that they want. And it's nice. It's why there's always a push for stem cells, because those are the important, amazing cells of the body. B cells mature in specialized bone marrow sites. T cells mature in the thymus. Both cell types migrate to separate areas of the lymphoid organisms or organs. So they just matriculate to the areas where they're most effective. B and T cells constantly recirculate through the circulatory system in the lymphatics, migrating in and out of the lymphoid organs, and of course, you know, it gets dumped right back into the circulatory system and eventually leak through the leaky venous system back into the lymph, and so on and so on and so on. So here is the overview of the function, and you can see the bone marrow and spleen and thymus and spleen. It looks where the migration of lymph and spleen. So these are different types of cells, where the B cell gets its name as bone, of course, and the T cell is thymus. And then antigen dependent responses do a fair amount of training, but the end result of the different B cell line and the T cell line end up with differences. And these are really clearly shown here. This is a great slide from the standpoint, you want the bird's eye view to the whole thing. Here it is. So the antigens are processed by phagocytic cells. So they go around and they grab cells that aren't supposed to be there. And then they kind of chew them up and then present antigens that could be potentials to create an immune response against, to clear them. And so those, now the B cells require stimulation from helper T cells. So you can see right in the middle here that, let me kind of point it out here. Here's the T cell, and if you notice the line goes across, the most B cells require stimulation from the helper T cells. So to get the ball rolling, here is sort of all of these cells on this side, the T cells, are the ones that make sure, they kind of check your ID. They're the ones that says, all right, have you got a badge to be here before we start any of this process? So they're the security in addition to knowing what's going on in the body. And so they give the authorization for the B cells to be stimulated, and then they activate to create plasma cells. Those are the ones that secrete different types of antibodies. We're going to talk about those. The antibodies are the things that go around sticking to things to clear them. And so the antibodies are a lock and key, and we're going to talk about that. The T cell line, we have the activated T helper cell, and this does the T helper cell is the communicator. Without this, which AIDS patients that actually develop AIDS, that is the target, what is taken out, and it stops the communication, and so that we can't develop helper cells and T regulatory and cytotoxic cells and even the memory cells. And all of these play a big role in the management of the B cell line as well. So it really, you want to talk about a key component to shut down an immune system, you go ahead and you take out the communications. And that would be it right there. Okay. I hope that makes sense. The chart's a good one. It's a figure. It's in your textbook. Pathogens carrying antigens cross the first line of defense. Resident phagocytes migrate to the site. The tissue macrophages ingest the pathogens and initiate an inflammatory response. Dendritic cells ingest the antigens, migrate to the nearest lymphoid organ, and they process and present antigens to the T and B lymphocytes. So these are sort of the paper shredders, and then they present the pieces and parts to ones that are the authorities that can actually do something with them, which would be the B and T lymphocytes. Gamma delta cells can be activated by these pre-programmed or series of proteins that we talked about that are presented in the membranes that are unique or common through bacteria and viruses and all those sorts of things. And it's sort of a fast path. Okay, we've seen these before. Let's go ahead and get your process, that sort of thing. So all cells have a variety of different markers on their surfaces for detection, recognition, and cell communication. So I have a little picture of a card reader over here, and it has a sort of information on this little card. That's sort of what's going on. The detection is, all right, what type of protein markers do you have? Do they follow the sort of types and styles that we see of this particular, so that would be the format of the sort of the card, and the reader is the communications part. And so the equivalent would be proteins that respond to other proteins and communicate within the cell. So that's sort of the function of the little reader there. And so it does, the analogy is pretty spot on there. So these markers play an important role in activating different components of immunity, but there is sort of a making sure that we know what's going on and we're not allowing bad players in the system. We've got to kind of screen. And the way we screen is using the major histocompatibility complex, which is a series of proteins that are unique for us. And there's a set of genes that code for these. They give rise to a series of sugar proteins, glycoproteins, found on all cells except red blood cells. And, oops, we got a problem here. Let me deal with that. Okay, so let's get rid of that. All known as human leukocyte antigens, or HLA. So these HLA antigens, of course, if you've heard of people that get, let's say, something, a liver transplant or something like that, they always worry about rejection, and that's always comes down to how close in the family do the HLAs or the MHCs match. If there is a slight difference, then we're going to have to deal with some incompatibilities. The more incompatible it is, the more serious it becomes in terms of dealing with that. So that's one of the concerns that we always have. These markers play a vital role in the recognition of self by the immune system and rejection of foreign tissue. So the recognition of self is really, really important. In other words, does it have this display that is sort of like when you go in to a bar or something, not that I do that, but you get your hand stamped, and so you can go in and out freely, and they already know that you paid the fees or whatever. That's sort of the analogy for recognition of self. And then others that don't have those stamps have to pay or do whatever it is, or they get cleared by the bouncer. Isn't that just a wonderful analogy? I wouldn't share that with anybody. 3 classes of MHC genes. Class 1, code for markers that appear on all nucleated cells. These are the class 1s are the sort of important for self molecules and regulation of immune. Class 2 encodes for the immune regulatory markers found on macrophages. All those that do the work of processing antigens and presenting them. And so the class 2 genes are sort of the, they're not invoked until the security check has been done. And they won't act to present unless they are processed properly. Class 3 genes encode proteins involved with the complement system. These are, once they get invoked or turned on, the series of switches, that's it. It's going to happen. And again, this is the domino effect. or cascade as it's known. So class 1 and 2 of human major histos, you can see class 1 molecules on all nucleated. Class 2 found on some types of white blood cells, and these kind of go together. It's sort of a part of the process of checking things. And then the CD molecules, the cluster of differentiation. That we have about 300 of them so far that we've recognized, and many of them, like the CD3, 4, and 8, are really important types of cluster of differentiation. All they are rafts of proteins that float around in the membranes that sole purpose is a sort of a unique fingerprint or a uniqueness of the cell, and it kind of identifies the type of cell and other features. So lymphocyte receptors' major role is to accept or grasp antigens in some form. B cells have receptors that bind antigens. T cells have receptors that bind antigens that have been processed and complex with the MHC. Only the T cells are complex with the major histos, okay, not the B cells. The B cells don't even get turned on until a T cell tells them, and the T cells have already seen these, they've been blessed and know that everything is okay. They've gone through the security check, and they're authorized to turn the B cells on, if that makes sense to you. Receptors have the capacity to respond to nearly an infinite number of different antigens. It's an amazing system. So when challenging B and T cells with antigens, both these cells proliferate and differentiate, so they do their job. A clone or a group of genetically identical cells is created. So we make a huge number of identical cells. Some clones are memory that will ensure the future reactiveness. So there are groups of sort of pliable or programmable entities that are going to provide or confer specificity. And both the B and T cells respond differently. As you already know, the B cells are more in the antibody humoral type of production of things, or in the blood, where the T cell is more cellular based responses, and you should pick that up. So T cells respond to antigens. This is the T part. The helper T cells activate macrophages, assist B cell process, and help activate cytotoxic T cells. These are the cells that actively go out and kick butt of cells that aren't right. We got to get rid of them. Regulatory T cells control the T cell response by secreting anti-inflammatory cytokines or preventing proliferation. So these regulatories are usually down-regulatories, and they're there to kind of squelch the immune response because the battles are already been ongoing, and it's probably a good time to curtail it, and the specific type of immune response should be winning by this time. Cytotoxic T cells lead to the destruction of the host cell. It's usually where a virus has infected a cell and it's been determined that that cell's got to go. Cytotoxic T cells do the job. Take it out. When activated, B cells divide and give rise to plasma cells, and that's the cells that secrete antibody. Antibodies attach to the antigen for which they're specific. And that's the thing, is that they bag and tag, or they tag and then get bagged based on that antibody sticking to it, and or it neutralizes so it can't bind to various things. Alright, so now we're going to shift gears and move into summarizing the maturation process of both B and T cells. We're going to look at how this is done, usually with a diagram or so, and then describe the structures of the B cell and T cell receptors. Then we're going to talk about clonal deletion. Okay, so contrasting the properties of B and T cells. And this makes it really easy. Let's start off. B cell, of course, bone marrow. T cell, thymus. Specific surface markers, immunoglobulins. The T cells are T cell receptors with those sort of the differentiation type proteins that are on the surface, the CD molecules. they circulate in blood, the B cells in low numbers, the T cells in high numbers. And they're the ones, remember, they're responsible for regulation, so they got to be out in force. Receptors for antigens, the B cell receptor, are those immunoglobulins, which is another fancy name of talking about antibodies. And then the T cell, which is similar, is a T cell receptor, which is sort of like an antibody embedded in a membrane, but it doesn't go anywhere. It's kind of cemented in. Distribution in lymphatics goes to the cortex and follicles. The T cells go in the paracortical sites, interior to the follicles. Those are the ones that, if they're regulatory, they get the fancy rooms, and they're sort of the control parts of the lymphatic organs. Required antigen presented with MHC. The B cells, they're told by the T cells that everything's cool, so no, they don't deal with that, but the T cells do, of course. That's how they verify. Papers, please. And if you don't have the proper papers, well, they just shoot you. There's no jury or anything. You're not supposed to be there. You won't be there, and that's kind of how it is. Product of antigenic stimulation, plasma cells and memory cells. These are the cells that are involved in the production of antibodies. And of course, we want the cells that produce them, so we're quick and fast on the draw there. The antigenic stimulation, several types of activated T cells and memory cells on the T side. Not to be confused, but they have their own backup. So the regulatory system has its memory cells, so they don't have to reproduce or reconstruct things. This saves time, working easier and smarter to get the job done. So the general function of a B cell is to produce antibodies, inactivate, neutralize target antigens, where T cells help other immune cells suppress, kill abnormal cells, mediate. It's all regulatory type of functions and works with the major histos to verify everything. The specific events in T cells, maturation of T cells in the development of the receptors is directly in the thymus and its hormones. CD3 receptors surround the T cell. B cells don't have this. The T cell receptor and assisting binding. The C4 co-receptor, accessory receptor proteins that helps the T cell receptor bind. It sort of supports it. It stacks these incredibly large protein and sugar type molecules, and it needs a separate support, and that's sort of what they look like to the MHC2 molecules. CD8 receptors or co-receptors are found on T cells and helps bind the MH1s. So the CD4s are 2 and the 8s are for the MH1s. And you can see here's an antigen binding site drawn here. It's a T receptor, and you notice it has the CD4s and the CD3s around that, and of course they emanate from the thymus, hence the T cell. And you can see the MHC1s, and kind of everything is in place as it's supposed to be. The phospholipid bilayer, and they're kind of bound into it. So you have the antigen binding site sticking out, and the FC, or the constant regions, what's buried in the membrane. Remember that. That's kind of as simple as the antibodies are like that, but these don't go anywhere. These are permanently kind of mounted into the membranes, and then these receptors help the communications and the specificity that they use for their regulatory purposes and to communicate with other cells. And the B cell development, of course, come from the bone, and they mature and they form pretty much memory B cells and plasma cells. But in between here, of course, we have to have communications with the T cells to make sure that the types of processes and productions that we're making are legit. And we've got to make sure all of this, so that the B cells are maturing through the bone, then they migrate to the lymphoid tissue and do what they're supposed to do. And the surfaces of these kind of look like this, where we have the antigen binding site on the surface of the B cell, which, if you notice, the MH3 markers and things aren't directly attached. They're not binding to it, they're just binding to the antibody. So these are sort of the normal things that we find in the surfaces of cells anyhow, and that's where it could possibly be confusing. But we don't worry about MHC with B cells, only with T cells. By the time B and T cells reach lymphoid tissues, each one is equipped to respond to a unique antigen. That's the whole purpose of this. Diversity is generated by a rearrangement of the gene segments of codes that code for a particular antigen receptor. Now this is what's unique. What we have are the coding regions that flip-flop. They randomly change around in these little short segments, and we got hundreds and thousands, 100,000s of these things all connecting and flip-flopping in certain ways that makes a unique variable region. And it's random. So it's the same if I were to give you a lock, and like a Yale lock, and I say, okay, here's the key blank that fits that lock, but it's not ground yet. So I want you, here's a couple 100,000 of these blank keys. I want you to randomly grind those keys until you get one that fits the lock and you can open it. Now that's a daunting task, but that's the immune system. And the way it does it, it has these random little processes that go on that flip-flop these segments of DNA. They create sort of a unique arrangement of these antigens here. They don't know if it's right, but it's random, and if it actually starts to bind to the antigen, hey, we've done our job. Let's keep that one, and we've done what we needed. So the video that you saw on antibody diversity should convey the genetics behind that process. It's unique, and the assortment arrangements, huge number. So that's the key. And so all these different variable regions, and I'm not going to ask you to know that, but there are certain regions responsible for the constant region, but we don't need the flip-flops there. We need to keep it constant, but the variable region, the antigen binding region, that's where we need to have that sort of arrangement. And to keep it consistent with our philosophy, here's all the key blanks. We got to grind those randomly until we find one that can open the lock. And that's quite a task. So the specific B cell receptor is the immunoglobulin molecule that we have to grind, and it's a large glycoprotein molecule that serves as the antigen receptors of B cells, and they have an antigen binding site, a variable region areas of extreme versatility, and then they have light and heavy chains, and I'll show you that, and the constant region. And so all that boils down to right here. Here's the heavy regions. These are the heavy chains right here. These are just the longer parts of the protein. They kind of bend out to form that V at the end, and then they get this little short piece, the light chain, that's bound by a sulfhydryl group. These are sulfhydryls, and they're pretty strong. If you ever get your hair permed, the hair is formed by sulfhydryl groups, or mercaptic type smell that we don't like, but the antibodies are held together by the sulfur type bonds. So the strength of these are similar to the permed hair, kind of bending the hair in a certain way. And so anyhow, so there's the variable region. The constant regions are down here. This is what sticks in the membrane. They call that the FC portion, and this is the AB, or the antigen binding portion, at this end. Now the antigen binding, of course, here is the key, here is the lock. So whenever there's a positive charge here, there has to be a negative charge here. In fact, I show the slide for that, but not only in charge, it has to form in shape. So the basic shape. So we have the antigen is in the green. This is the antibody. So wherever the antigen has a negative, we got to have a positive on the antibody side, and a complement type of hydrogen bonding perhaps that will work. A van der Waals force is if you ever take a helium-filled balloon and rub it on your hair and it lifts your hair up, assuming you have hair, I don't, but you rub it on your hair and your hair picks up, that's a van der Waals force. That's the strength of a van der Waals. remember, individually weak but collectively very strong. So these collectively can be quite strong, and so they bind. The antibody, the Y portion here, we call that the hinge region. Here's the FC region, but that's where it binds the antigen. But it's not just binding it; it has the shape and that randomly had to be configured, and the charge. So the antibody had to have a complement charge and features randomly come across by the flip-flops of that genetic system, which is quite amazing. Let me zoom in just a little bit more, and you can see the interactions of the arrangements of particular amino acids by the flip-flopping and arrangement. So we have electrostatic, hydrogen bonds, van der Waals forces, and hydrophobic forces all contributing now to the antigen-antibody arrangement. You see that? So you can see the red part and the blue part. Those had to be compatible, shape-wise had to be compatible, the way that folds and all those things. And then we have the charges, an ionic bond. Here's a positive charge and a negative charge. So there's the antigen, the antibody. So really quite amazing that all of this happens. We're going to talk about, there's some vocabulary words in here. We've already talked about some of these, and you've had these in other courses. But a paratope. The paratope is the region on the antibody, and we're going to talk epitope is on the antigen side. And so what's in red here is the epitope, and you can see the other, what was going on here? The slides decided just to go on their own here. Go ahead and hide that. Okay, so epitope, and what was the name of this one? Paratope. So it is similar to it. Okay. T cell receptors belong to the same protein family as B. Similar to B, they form genetic modifications, have variable and constant regions. It's sort of like just stuck in antibodies, but they're inserted in the membrane and they're kind of glued in. That's my own word. They're not really glued. They're just attracted by charge. The mechanism by which the exact correct B or T cell is activated by any incoming antigen. The lymphocyte specificity is pre-programmed and existing in genetic makeup before the antigen ever has entered the tissue. Each genetically distinct lymphocyte expresses a single specificity. So when we have a clonal expansion, we push the button of the one that actually works. And that is a clonal expansion of the antibodies that actually can recognize the antigen. And then we multiply those as fast as possible. We're mobilizing the troops, and they all have the specific weaponry for that one particular troublemaker, and only that one. One potentially problematic outcome of random genetic assortment is the development of clones that can react to our own tissues. This can happen, and why some people suffer from various types of autoimmune diseases, because part of the system's not working properly that does the clonal deletion, and then they suffer from various joint damage, all sorts of various things, and type 2 diabetes or type 1 or whatever. And it's usually due to autoimmune type of issues. And so clonal deletion is important to get rid of any of the troublemakers that are the resulting of this random assortment that's gone on. So here's a kind of a diagram. We have all sorts of assortments, and here's the one that actually is the winning. We need to get rid of these. We don't want them, so we got to delete those out of there. But these are the ones, and here's the epitope that bind to that specific antibody, and then we just now proliferate and make those antibodies, because those are the winners. The other ones, it's great that they were there, but we got to get rid of the self ones that bind. It's sort of like the keymaster in the Matrix. The keymaster is grinding keys randomly for the event they have to open a particular lock, and when they find one that does, they stores it on the wall. Isn't that an amazing analogy? And all these, the body kind of collects all the various keys and immune responses that they've known over the years, and can come back and pick them up instead of regrinding randomly. Now we know which one is which there must be a huge identification. Part of the T cell work there. The T cells know where each of these are parked and pulls them out and rapidly deploys them. And so I have a diagram that hopefully it makes that analogy work for you. The antibody is the lock and the antigen is the key, and it has to form in shape and charge to fit. And so here's the other part of that, but that's the hypervariable regions, and these are done randomly until now in memory we can just pull the lockout when the key starts to show up. And there you go. Again, just reiterating, the complementary determinant region is where you grind the key, and it fits the antigen by charge and shape, the way the sort of the analogy is for the antibody there. So so here's a bunch of random keys that have to go and be ground randomly. Just randomly grind them up, and then all of them we quite check the keys to see if we can open the lock. We got one out of all these keys that works. We got to put that in memory. But meanwhile, we don't want to keep these other keys that we've ground, because they may open doors we don't want them to open, so we got to get rid of them. So that's the clonal deletion. But we take that one key, that one right there, and that one we're going to amplify and keep in memory, and we can pull that key out just like the key master when it's stored on the wall. And when that time comes, the T cells will say, all right, I know exactly where that key is, pull it out and let's get going and build the response. Characteristics of the antigens that optimize their immunogenicity. Describe how the immune system responds to these alloantigens. Some more vocabulary. Alloantigens, super antigens, and allergens. Let's get through that. This is really not hard. To be perceived as an antigen or imagen, a substance must meet certain criteria: the foreign shape, size, and accessibility. Whole microbes or their parts, cells, or substances that arise from human plants and other types of molecules. Complex proteins and protein-contained molecules are more immunogenic than repetitious polymers like sugars, polysaccharides, and things like that. So, antigen categories: proteins, polypeptides, enzymes, cell surface structures, exotoxins. Lipo, fat proteins, cell membranes. Glycoproteins, blood cell markers. Those are glycoproteins for the A and the B, or the AB, or not have any markers, the type O. Nucleopore proteins, DNA complex to proteins, but not pure DNA. So they're complex with proteins. Polysaccharides, certain bacterial capsules, poor antigens, just really poor. Lipopolysaccharides, a kind of sugar fat type of composition. I gave you the antigenic determinants epitopes video. It's actually very good, and hopefully it will be helpful for us to communicate with this presentation. Good antigens, or characteristics of good antigens: they're large, and their size alone is not enough. A glycogen is highly repetitious structure, and molecular weight over about 100,000 Daltons. Water is about I forget, it's about 15 Daltons. That's it. Water is just 15 Daltons. So you can see the size, just the water molecule. These are considerably bigger. So insulin at 6,000 Daltons. It's a measure of weight, molecular type of weight in a Dalton. You can look that up. So you can see protein epitopes. The epitopes are the site where the antigens are they're actually the docking, or the part of recognition of an antigen is the epitope, and we'll talk about that here in a minute. Microbial cells, plant molecules, all of these are pretty good immunogens. Poor immunogens are the trisaccharides and the polysaccharides or the polypeptide, just pure ones on their own. Anything repetitive is really tough for it to recognize it. So an epitope is an antigenic determinant, are the portions of the antigen that are responsible for the specificity. So there's the portion of the epitope that the antibody actually binds to. And I just wanted to make sure that was clear. Your book was a little confusing about that. So here is the epitope. There is the paratope. This is the complement. So the epitope is the antigen. The paratope is what matches the shape and charge, the opposite charge, to the antibody. So we call that the paratope. Now, an antigen could be a protein, could have more than one epitope. It could be covered with various different epitopes, and different antibodies could be raised to those epitopes. And I just wanted to point that out. So here's a series of 3 different epitopes on an antigen. So there you go. It's the same sort of thing. That sounds confusing, but it's really not, because it's driven by the epitope. And you can see having the various sites and various things, and antibodies binding to them, the epitopes. Haptens are different from the standpoint, if the antigen is really, really small, we can enhance it by this concept of a hapten. So smaller components linked together with some sort of carrier molecule, we get a complete antigen, and that's called a hapten. Okay, so hapten molecules with a carrier, these are carried together to create the immune response. So these haptens happen to bind, if you want to remember it that way, to a carrier molecule. So the haptens happen to bind to a carrier molecule, and then that helps facilitate the immune response. Examples of haptens are drugs, metals, ordinary, innocuous, household, industrial, and environmental chemicals, but they get bound up with other types of carrier molecules, such as serum proteins, and then they become better immunogens. And that's why a lot of these things like sprays and dusts and perfumes can cause your nose to gush and create a immune response that's causes the histones to the histamines to be produced, and then you need tissue paper, and it gets nasty. Alloantigens, surface markers of molecules that occur in some members of the same species but not in others. So allo just means it's sort of different within a group, and it's the same for the individual blood group, and I have an example for that, responsible for incompatibility. So we all know, maybe you do or don't know, I'm A positive, that's my blood type. So antigen A has a particular protein on the surface, antigen B if you happen to be type B, or if you're type AB, you have both of those on your red blood cell. If you don't have any expressed, then you're type O. So O is a universal donor, and AB is a universal acceptor, as a recipient for the blood types. You got to be very specific. But you see, the antibodies, if you are type A, then anti-B would be present, and things like that. And then they agglutinate because the antibodies can cross-link. There are different types of antibodies, like IgM and those sorts of things that can do that. And we know the blood types, it's a really crappy graphic, but gets a point across, anti-A, anti-B, anti-D. So you can see the clumping as a result of what I just showed you, the clumping because the cross-linking of the antibodies causes clumping effect. So we know anti-D is the Rh factor. I didn't mention the Rh factor, but they call that type D. So this would be O positive because it didn't have the reaction, and but it did have a reaction to the Rh factor, so it would be type O positive. O negative, they're all negative. A positive, like myself, have clumping for the anti-A, so it picks up the A antigen, but not the B, and then of course the Rh factor, so that's type A positive, and so on and so forth. And then you can see everything kind of follows for those. But those are examples of alloantigens. And what's different about type A and type B, if you ever were curious, it has similar structure. You can see N-acetylglucosamine and galactosamine and those various colors. and then you see the A antigen has a galactosamine at the end, where the B has a galactose. Type O doesn't have any. So there's the epitope for that particular antigen right there, or lacking of one, and so you can see that. So in case you're ever curious about that. Superantigens. Bacteria toxins that are potential stimuli for T cells activate T cells at a rate 100 times greater than ordinary antigens. The result can be overwhelming release of cytokines and cell death. Toxic shock syndrome is a certain autoimmune disease associated with superantigens. So superantigens evoke an allergic reaction, and what they do, I have a graphic just really quick. What it does, it sort of cross-links what normally wouldn't have been a connection, and the superantigen sort of cross-links and causes the presentation and triggers the T cells to do things. And because it's such a confusing one, I wanted to make sure that you saw the video, and that hopefully you've seen this, and it talks about superantigens and how this is presented to T helper cells, and that sort of gets the ball rolling there. Antigen processing presentation. Most of the immune reactions to antigens must be formally presented to the lymphocytes by antigen presenting cells, APCs. Examples of these are the macrophages, B cells, and dendritic cells, which we've already talked about. So that's really just a rehash. And I had a presentation based on the antigen processing that should help with that. And what the videos talked about are how antigens are presented to T cells. And so we have microbes that are digested by the it's been engulfed by an antigen presenting cell, and then kind of chews it down, and then spits out peptides or pieces, Reese's pieces. No, that's just a joke. Peptides or pieces, and the antigen pieces are complex with MHC2. So in other words, it sort of gets the validation for your system by the major histocompatibility, transported to, and then it can go on and now later communicate this to other cells with the processed pieces and parts. Because anytime something becomes activated, as you know, it's going to cost the cell money and resources. And so once this happens, the cells communicate with these receptors, validating it, the T cell receptor, and then becomes activated and then assists with the B cell. So it's turning on a whole group of people, and before you turn on the resources, we've got to validate it, and that's what the MHC does, and that's the purpose of the T helper cell, is just to make sure that we don't randomly just turn on these processes and start causing a mess. It has to go through the proper authorities, and that's the way it's done. Antigen processing and presentation. Most antigens must be presented first to T cells. We just talked about that. I just want to re-emphasize. I try to make it sound like I was saying something new, and then you realize, well, you just said that. That's why I do that. It's redundancy. Builds good memory. Even though they will eventually activate both T and B cell systems, the T cell independent antigens can trigger the B cells directly. But the thing is, it does it with a major histocompatible, so it's been authorized. It's sort of blessed, you can kind of say. Carbohydrates with many repeating and variable determinant groups, like polysaccharides, things like that, are just not very good with creating an immune response. So sometimes even those can be triggered. So you can see the antibodies sort of stuck in the membranes of the B and T cells. This is permanent, and the B cell, that's the purpose. It's going to create these plasma cells that's going to secrete this particular type of antibody. This doesn't do that. This strictly is an association of binding and communications. And I have a fancier drawing than that if you like that sort of thing. But you can see that the CD3s are associated with the T cell and the major histos and all that, where on the B cell, now it's kind of just sitting there doing its thing, and they have the same paratope between the two. Of course, that was the one that was used sort of as a model. My system's telling me that I'm going to be past the limit of YouTube if I don't shut up. I didn't quite get through the whole slide set, and that's okay. So that means we will have a part 2 in this, and which is just fine, because I get into vaccines towards the end, and I have yet one more video, and I'm going to tack that on to part 2 of this video to kind of round you out. So I hope you've enjoyed this to some degree. It should be helpful. If not, I hope you got what you needed out of the material.